Thank you very much, and good morning, colleagues. It is exciting to be here. I hope my slides can come up. It's exciting to be here and speaking to you about HIV vaccine clinical trials pipeline. We're not discussing funding, my bio changed, and many things happened. But, so we'll talk about the science, because the science still remains. Penny has done the big part of it. I'm going to just share some of the trials that have happened using some of the strategies that she's presented. Some may be going on, some may not be going on. Disclaimer, please check the website, speak to those doing these studies, and be clear on where we are at with them. But I'm confident that since the science remains, we can pick ourselves up and continue to work on these trials. So I have no conflict of interest, but I also wanted to welcome you to Kampala. Those of you who have not been there, lovely city, beautiful sceneries, and lovely people. So the presentation outline, I'll do a bit of what has been done already, and then hopefully we'll do some justice to the trials pipeline. The statistics, every presentation yesterday discussed it. We still have lots of people infected with HIV, we still have people dying, and we still have many people that are living with HIV and need care, need treatment. But these statistics is the real reason we actually need a vaccine. We know that many infectious diseases have been eradicated because of having vaccines. And for us to ultimately end HIV, we really still need a vaccine. The medicines are helping, everything is, the prevention is helping. Now with all that is happening, we wish we had a vaccine sooner, then would not have to worry about all that is going on. So we still need to work hard to find a vaccine. The challenges Penny has elaborated remain. But we remain focused as well on the work we're doing to find a vaccine. So she's talked about this. Right now, the biggest challenge, or the newest, not the biggest maybe, but the newest challenge is the funding cuts, because that is taking us a lot more slowly than we want to go. Lots of the programs have been cancelled, and that means we're going to have to go backwards years of research and progress are going to be hindered by this, but everything else she has mentioned as part of her talk. Lessons from the previous studies, as you can also see, I've also highlighted AMP and the lessons we learned about broadly neutralizing antibodies, and where the science is going now is really informed by many of these, and she has really elaborated on the science as it is moving. So some of the strategies, germline targeting. So I'll try and explain some of the studies that have been going on. The multiple founder variant strategy is also one of those where trials are being planned, or some have already started. There are many trials that are now trying to use novel viral vectors to try and move immunogens into the body and cause immune responses to happen. So we have some gorillas, adenoviruses, and CMVs, but also newer adjuvants are being tried to see how best we can optimize these vaccines. And then all the many delivery platforms. She's just told us about the mRNA platform, their nanoparticles, their ferritins. So this talk is nowhere near exhaustive, but will be used to tickle our minds to see how we're moving. And then we have a trial that is testing how to administer products with the assumption, or the assumption that if we mimicked acute HIV infection, we might be able to induce better immune responses for people without HIV. So I'll discuss some of those. So the vaccines are targeting different parts of the virus, the epitopes, CD4 binding sites, the trimers, V1, V2 sites, and V3, all in the trying to aim at producing broadly neutralizing antibodies, and hoping that these broadly neutralizing antibodies will help us target several strains in the end and cover us more than before. But also there are studies that are using immunogens that have been developed, targeting conserved regions of the virus, those regions that do not change much strain to strain, between the different strains, and hoping that because they are conserved, there's likelihood that they will give us immune responses that will cover a broader range of the strains and be able to give us a broader cover. So currently, broadly neutralizing antibodies is a focus, like Penny has nicely alluded. But we also know that we're probably going to need a combination of different vaccines. So T-cell vaccines tested in the past did not work, but we think it's probably because of the way they were designed. We still are picking these up and thinking that we might need the antibodies as well as the T-cell vaccines to try and cover us for a vaccine. So a combination approach is required. So it's not lost. There are members still working on finding a T-cell vaccine. But also for all vaccines that we test as part of the immunogenicity testing, we look for antibodies, we look for T-cells, we look to see how good they can do, across the board. So the pipeline I'll go through includes some of the studies that have happened. Caprisa, she's happily explained that. She's part of it. She's done a lot of work on these antibodies. Germline targeting, IAVI has a pipeline, and many other people, so this is not exhaustive again. Then we have the trimers, then we have the sequential slow delivery from MHRP, T-cell vaccines, multiple founder variants. Brilliant, very close to our hearts. Unfortunately, the grant was cancelled, but the science remains. And hopefully, Linda Gale here, who was our PI, is trying to see how we can actually run the Brilliant 001 trial in minimal ways, depending on the funding that comes through. And then there are studies on novel vectors. If I don't get to go through all of them, we shall see. So there's this Caprisa trial program. Some of these studies are completed, but these are things for us to watch out for. So I chose to speak about Caprisa 012B, which tested both IV and subcutaneous administration, open label, but also randomized, all in one study, different doses. And this CAP256 antibody now engineered for clinical testing, but it was tested in combination with other antibodies. So it's not just one antibody. We think that different antibodies would be needed to be able to broaden the target for the different strains, because even one individual has several strains of the virus going around in their body, so one might not be able to. The AMP trials showed us that some very rare people were able to have antibodies that were sensitive. The antibodies that they received were effective or efficacious, but to very specific strains. So this means that if we target, if we use combinations, we might be able to target several strains. And these are targeting different viral epitopes. And this trial, 012B, either when the CAP256 was administered alone in combination with another VRC07 antibody, it was safe, favorable pharmacokinetics, and there was neutralization activity. But it also demonstrated that on top of IV administration, subcutaneous administration was possible. What this means is we need to follow the Caprisa pipeline and see how far that takes us. Because a lot of work is still going on. And then IAVI has these pipeline trials. I chose G001, which is already ended. G002, I think, also ended recently. But they are testing this germline targeting immunogen. They did a phase 1 trial, which is the first in human trial. And the immunogen EODGT is a nanoparticle vaccine. And it was randomized, placebo-controlled, and it was targeting to measure precursor B cells. Like Penny has indicated, for germline targeting, you induce precursor B cells, but we are going to need subsequent booster immunizations with whatever, maybe either broadly neutralizing antibodies or T cells, whatever, that is going to help those precursor cells actually mature to the target broadly neutralizing antibody that we need. So there's a long way to go. They tested different doses, but what they showed is that this product was able to induce VRC01 class memory B cells, and also induced some CD4 T cell responses, and the T cell responses were, of high magnitude and highly polyfunctional. But IAVI has a pipeline now. This product being utilized using the mRNA platform, or developed using the mRNA platform, and I think the middle one is the IAVI G002, which has also concluded, but I couldn't, give you all the information about it. You can read about this pipeline. And in 002, they have tried to combine this germline targeting product with a booster product that is aimed to mature the B cells, the precursor B cells that would be produced. So IAVI has a pipeline we can all learn about. I know they have also been impacted by the funding cuts, but like I said, the science remains, and once collaboration and the spirit to keep moving is on, hopefully we'll be able to move this science forward. Like we said, T-cell vaccines, Penny also mentioned these. T-cell vaccines are still being looked at, and there's this lovely trial that is done by the University of Oxford, and this trial has, the immunogens have targeted conserved regions of the viral envelope, Pol and GAG, and they are intending to develop T cells, T cells. So it's a T cell vaccine. So when they gave it out, they gave it to HIV negative people. They gave a low dose group and a high dose group, and then they gave a combination of the products. And then they gave one with the chimpanzee adenovirus as the prime, and then the immunogens in the MVA vector as boost. And then found that the one that received the high dose with the vector, with MVA as vectors, elicited robust T cells in all the volunteers. And this T cell immunogenicity was good, polyfunctional, and was capable of proliferation. This study is interesting because this product is actually being used as part. They're going to try it, or they've probably started some trials to test it in people who have HIV. I think this is one of what Penny has said. How do people with HIV actually, how do these vaccines, how can they help in people who are already virally suppressed? So this T-cell vaccine, we need to watch out for those ones being given in people who already have HIV, to determine, its use as part of a cure research strategy, but as a therapeutic vaccine. Again, there's many other T-cell vaccine work that's going on, so we can look these up and be more informed in that area. So the multiple founder variants, this is being done by MHRP. Penny has mentioned that there's actually people, maybe rarely, but who get broader neutralization after years. And one of these was a Kenyan participant that MHRP is using to optimize sequences from that participant and then develop a vaccine. They were producing more broadly neutralizing antibodies compared to those who had one founder variant. And we're testing another study. This is a study at my site actually currently going on, where we are mimicking acute HIV infection. So the design is that we are giving products one, in the normal prime boost regimen, but the other where we're giving some of the participants increasing doses of the immunogens, mimicking HIV replication cycles at acute infection, to determine if that mode of administration would eventually help to produce better immune responses. So this one is ongoing. Let's watch out for it. Lots of pipeline work using ALFQ as a new adjuvant. There are many adjuvants that have been used. This one is still being tried. But also the multiple founder variants vaccines are currently being developed to be tested in humans. HVTN we all know HVTN has had a huge history of HIV vaccine development. And as part of their pipeline, they had these 2 vaccines trials that were being planned to start in South Africa. And I learned this morning from Linda Gale Baker that HVTN 317 may be able to proceed, which is great news in one way or the other. I think there's challenges, but it was supposed to have started recruiting this month, but there's hope that it will continue. So let's watch out for these. It's exciting to know that some work can still continue. And then Brilliant, I cannot not speak about Brilliant. It's so close at heart. So we were planning to actually start a phase 1 study in January. And we didn't get to start. All approvals received. But we're also going to test a germline targeting with 426 BG505, which is germline targeting with 426, which is also one that looks at VRC01 epitopes. And we had a whole lineup of development program, experimental trials that we're planning to do. Again, Linda Gale tells me that in one way or form, this Brilliant 01 is going to be modified, but it might run. So let's watch this space and see what comes out of there. So what is the takeaway message? We still don't have a licensed vaccine. The science is there. The science is promising. There's still lots of work to do and challenges. And right now, we don't have anything close to phase 3, which means we are still far in the development plan for us to get an effective vaccine. Collaborations between industry, our governments is going to be critical for moving us forward. There are therapeutic vaccines, which I have not discussed, apart from the T-cell vaccine I have mentioned. So you can read about therapeutic vaccines and how those can happen. There was a vaccine trial that was planned in children under HVTN. I don't know which one that is, but maybe if Penny can tell us when she gets questions, she can add that to her update. We need sustained funding. We discussed AI and machine learning yesterday. It was exciting. For epitope designing, we need support to continue innovations with the vaccine platforms, personalized vaccines. We're all different. Penny just told us, many of us will produce different responses if given the same vaccine. And we also need to really educate our communities about these. So I acknowledge everybody whose work I have utilized today. And of course, Interest Workshop for giving me the opportunity and the podium, and Makerere University Walter Reed Program for allowing me to be here. Thank you very much.